Good morning, everyone. I'm Lorena Benedetti, and I'm a postdoc in the lab of Jennifer Lippi-Conshworth at Gianiglia Research Campus. First of all, I would like to thank the organizer of this cell biology virtual event for the opportunity to present my work here today that has been focused on the engineering of optogenetic tools for the manipulation of protein localization and signaling at the intracellular level, and in particularly at organelle contact sites. I would like to start my talk today highlighting the importance of having a tool to precisely control protein localization at the subcellular level. Why do we need such tool? The answer is very simple. The physiological function of proteins within cells is critically dependent upon their subcellular distribution and local regulation. Therefore, if you want to really understand the role of enzymes inside of a cells, we need to be able to redistribute this protein inside of the intracellular milieu with the same type of spatial and temporal precision. However, how can we localize the activity of a protein in a confined portion of a cellular subcompartment? How can we deliver a protein to a single intracellular organelle? And even more, how can we temporally control the duration of its activity in the area of interest? In the attempt to address these questions, we look into the methods that would enable us to manipulate protein expression and localization. Genetic manipulations like RNA interference strategies or genome editing strategies are extremely powerful tools. However, they occur on a relatively slow timescales relative to protein dynamics of intracellular signaling networks and any post-translational physiological process, and they lack of spatial control at the intracellular level. Chemically inducible dimerization tools, like those based on FKBP12 or its interacting partner FRB in the presence of rapamycin non-analogs, are extremely versatile tool for the acute control of protein recruitment to intracellular compartments, but also in this case, the lack of spatial control at the level of subcellular compartments. While light-inducible dimerization tools are perfect tools for this purpose because they enable protein perturbation on a timescale consistent with the reversibility of intracellular reaction and with very high spatial precision. So during my talk today, I will first discuss how we can use optogenetic tool to control protein dimerization in subcellular levels. And then I will introduce our recently engineered enhanced magnet system as a powerful tool for this type of perturbation. And then I will show you some of the applications that we develop using this EMAG system in cell biology. So optical photoswitches are a powerful class of tools that enable to control protein dimerization with light. And the basics of these tools is the interaction between two proteins or domains, where at least one of the two proteins is a photoreceptor, that upon light radiation undergoes a conformational change, and in this conformation combines with interacting partner. The duration of the interaction between the two proteins depends on three main factors: the switch on kinetic, the duration of the photostimulation, and the switch of kinetic that define for how long the two proteins can interact one to each other when the excitation light has been withdrawn from the system. This slide briefly summarizes some of the most widely used optical heterodimerization systems used in cell biology so far. As you can see here, they can be broadly classified in blue light-dependent system or red and near-infrared light-dependent systems according to the wavelengths of light that are used to activate or inactivate the photoreceptor. While using red and near-infrared light-dependent system, we can confine protein dimerization in small fraction of the cell by temporally irradiating with activating light the region of interest inside of the cells, and with inactivating light the remaining portion of the cell. With the blue light-dependent system, we don't have this advantage. However, we can use microscope setup that are more simple and easily available in every department. So I started my work asking, how can we spatially confine dimerization without the possibility to control photoreceptor deactivation exactly using blue light-dependent system? And I decided to test the capability of these tools to confine protein dimerization in fraction of subcellular organelles using the endoplasmic reticulum as my testbed. And I chose this organelle because it's a very broad intracellular compartment that span the whole cell and where intrinsic membrane protein are extremely motile. In all my experiments, I expressed the two components of its dimerization pair in the same configuration. So one of the components is expressed on the outer membrane of the endoplasmic reticulum, and I will call this molecule bait, while the other protein is soluble in the cytosol and fused to a fluorescent protein that enables me to visualize this reporting during my imaging experiments, and I will call this molecule prey. There are three main factors that are critical in the capability to confine protein dimerization with these tools, and which I would like you to pay attention during the next part of my talk. First of all, the location of the photoreceptor in the dimerization pair, so whether it's soluble or tethered to the membrane of the intracellular compartment of interest, the switch on and off kinetics, and the capability to localize the available prey for interaction in the area of interest. The first pair that I tested for this application is the one based on the photoreceptor cryptochrome 2 and CIB1 of Arabidopsis thaliana. In this dimerization system is characterized by a very relatively fast switch on kinetic with an half-life of about 4 seconds, and a relatively slow switch of kinetic with an half-life of about 5 minutes. And with this system, I tested the pair where the CIB1 protein is expressed on the membrane of the endoplasmic reticulum, while the photoreceptor, that in my schematic is always highlighted by a yellow box, Cry2 is soluble in the cytosol and fused to a fluorescent protein. These two images here at the bottom of the slide show you the distribution of M-cheric Cry2 before light irradiation. And as you can see here, the protein is diffused in the whole cytosol of the cell. On the right panel, in the whole cell activation part, you will see the effect of the irradiation of the whole cell with blue light, and it will show you how in this condition we can recruit the protein on the whole surface of the ER, while the central panel will show you the effect of the local delivery of blue light in a small area of 3 by 3 micron inside of the cell, with blue light pulses every 2 seconds. As you can see here in this image, using this system, we cannot confine protein dimerization only in the small area of light radiation, as indicated by the accumulation of the fluorescence in a broader area compared to the light area of radiation. And this is visible because in this case, I use a lookup table where the fluorescence signal goes from white to black when the accumulation of the protein increases. So in this case, the recruited probe goes outside of the area of lightivation for two main reasons. First of all, because the photoreceptor is soluble in the cells, therefore, as soon as it's activated in the area of interest, it can diffuse outside this area and interact with the available binding partner everywhere inside of the cell. And the second reason is because the switch of kinetic of the system is relatively slow, and the photoreceptor can remain in its activated state for a prolonged period of time, preventing us to achieve a very high degree of spatial confinement. In the attempt to identify a more suitable tool for this type of application, I decided to test the eyelid system. This system is based on the interaction between two E. coli peptide, SSRA and SSPB. SSRA is a very small alpha helix that has been fused on the C-terminal portion of the photosensitive domain LOF2 of a vena sativa phototrophin 1. This LOF2 domain is a perfect photoswitch because upon light radiation, it undergoes a dramatic conformational change with the exposure of this C-terminal alpha helix and thereby exposing SSRA to its binding partner SSPB. This system has two main advantages over the previous one. First of all, there are several dimerization pairs with different switch of kinetic, and some of those much faster compared to the previous one. And in this case, we use a system with a switch of kinetic with an half-life of about 30 seconds. And the second aspect is that this system is functional even when the photosensitive domain, LOF2, is tethered to the membrane of the intracellular organelle of interest. And in this way, diffusion of the photosensitive element is more limited because of the association to the membrane. So here again, you see two images showing the distribution of the soluble component of the dimerization system diffusing the whole cytosol of the cells before light irradiation. On the right side, you will see the effect of the irradiation on the whole cell, and in the central panel, the effect of the local delivery of blue light. As you can see here with this system, we can achieve a higher degree of spatial confinement in a fraction of the endoplasmic reticulum, but still this dimerization is not completely limited to the area of light excitation, because also in this case, the photoreceptor can diffuse outside of the area of interest and find its interacting partner that is always available for interaction. I finally tested the magnet system. And this system is interesting because not only it has the same characteristic and feature of the eyelid system with a switch on and off kinetics in the same order of magnitudes, but because this is the only optical dimerization tools where both components are photosensitive elements that must be co-temporally activated by light to achieve dimerization. So I also generate in this case a dimerization system for recruitment of a soluble protein to the endoplasmic reticulum. And as you will see here on the right, there is the effect of the whole cell activation of the system, so with the recruitment on the surface of the OLR. And here is the center, the effect of the local delivery of blue light pulses in this small region of interest. And as you can see here, with this system, we can actually confine protein dimerization essentially only in the area of light radiation. And this is possible for the fact that since also the soluble prey must be co-activated by light to achieve dimerization, we can actually confine the amount of available prey specifically in the area of light dimerization. And as soon as both components exit this area of light activation, they are inactivated. So from this first part of our work, we could conclude that all the 3 dimerization pairs tested are equally efficient in the modulation of biological processes upon light-dependent activation of the whole cell. And the switch of kinetic is the feature that defines the duration of the intracellular reaction. And this is the parameter at which everybody should pay the majority of the attention when choosing a pair for a specific application, because the switch of kinetic should match the dynamicity of the intracellular reaction that you would like to regulate using these tools. If you want to use one of the blue light dimerization system to confine protein dimerization in small subcellular volumes, you should choose a pair with fast on and off kinetics, where the photoreceptor is functional when tethered to the membrane of the intracellular compartment of interest, and ideally where you could control the amount of available prey for interaction in the area of interest. So from our experiments, the magnet system proved to be a very powerful tool for the light-dependent control of protein dimerization in subcellular organelles. However, their use in mammalian cells is limited by 2 main drawbacks. First of all, this photoreceptor requires a pre-incubation at 20 degrees for several hours that complicates its application in mammalian cell lines and prevents completely its use in mammals at all. The other drawback is that the dimerization efficiency of the individual photoreceptor is relatively low, so we need to use multiple units of this photoreceptor in order to elicit significant dimerization. So we decided to team up with the lab of Lauren Luger at Yale Research Campus in order to engineer an enhanced variant of this dimerization system in order to create an ideal tools for application in cell biology and ultimately in mammalian models. So the magnet system was engineered starting from the Neurospora crassa photoreceptor vivid. This photoreceptor is extremely small, is only 150 amino acids, and comprises 2 domains, a PAS core that binds flavinadenine dinucleotide, that is the photosensitive moiety of the photoreceptor, and an N-terminal cap that is the region responsible for the dimerization. In Neurospora crassa, when this photoreceptor is excited with activating light, in the N-cap domain, there is the exposure of a stretch of hydrophobic amino acids that mediates homodimerization of two vivid photoreceptors. The Sato group further engineered this dimerization tool by introducing in one of the components positively charged amino acid and in the other component negatively charged amino acids, thereby generating an heterodimerization system. In order to improve this dimerization tool, we decided to combine structure-guided protein engineering and cellular assay. And in particular, we used an assay to test all our magnets variants, the capability to recruit soluble protein to the membrane of to the outer membrane of mitochondria. So this movie was a beautiful example of our experimental assay, where we could see that before light irradiation, the soluble probe was diffusing the whole cytosol of the cells, and when the light was turned on, we can see the dramatic association of the soluble protein to the mitochondrial membranes. And when the light is turned off, we can see the rapid release of the probe as well. So this type of experimental approach enabled us to make sure that we were preserving all the positive aspects of the magnet system, so low background before light stimulation and efficient and fast interaction upon light stimulus with no formation of homodimers and fast heterodimer dissociation following light offset. But at the same time, we could select magnets pairs with new positive aspects. And first of all, we optimized the dimerization interface in order to increase dimerization efficiency and increase the thermostability of the probe in order to have a tool that was capable to function at 37 degrees. And for this type of mutagenesis, the strategy that proved to be the most efficient was based on the introduction of mutations that we identified in vivid analogs that are thermophilous ascomycetes. So there are ascomycetes that preferentially live at 50 degrees. So as you can see here, after our engineering strategy, we have been able to identify several magnets variants that you can see here highlighted in magenta and orange. They were characterized not only by the faster on and off kinetics compared to the original magnet system, and they were totally capable to dimerize at 37 degrees without the necessity to pre-incubate at 28 degrees, but also they were characterized by a 4 to 5 times higher dimerization efficiency compared to the original magnet system. So to summarize briefly some of the key features of our enhanced magnet system, first of all, is a tool that doesn't require an exogenous cofactor to be functional. Both components are extremely small and only 150 amino acids. It doesn't homoligomerize or form aggregates upon light irradiation, and both components can be fused at the N or C terminus of a protein and are functional when tethered to the membranes of intracellular organelles. We have engineered several pairs with different suites of kinetic, and this depends only on one component of the dimerization system that makes this pair very versatile for application that range of different temporal timescales. This system has a very low interaction in the dark, given the fact that both molecules are photoreceptor, and with this system we can achieve a very high spatial confinement of dimer by using a single wavelength of light activation. But let's see what type of application we can develop by using this tool. So as I mentioned before, these tools is ideal to recruit soluble protein or enzymes to the surface of intracellular organelle. As I showed you before, we could use this tool to recruit proteins to the surface of mitochondria, but we wanted to expand the palette, and so I generated tools that enables me to recruit protein to the surface of every intracellular organelle. So in the next movie, you will see how we could use this tool to modulate protein recruitment on the surface of the endoplasmic reticulum. And in this case, you will see how we preserve the capability to confine protein dimerization in a small fraction of light illumination inside of the cell at the level of the endoplasmic reticulum as well. This system is so powerful that we can recruit proteins not only at the level of broad intracellular compartment, but also at the level of the single individual organelle. As you will see here in this movie, we expressed in primary neurons a bait protein on the level of lysosomes, and then a reporter, LAMP1 RFP, to visualize the lysosome inside of the neurons. Simply delivering focal light at the level of the lysosome of interest, we can recruit the soluble prey only to the organelle of interest. But can we, besides recruiting soluble protein, can we really regulate intracellular enzymatic reaction inside of the cells? And the answer is yes. So I was interested in developing tools that would enable me to modulate phosphoinositides at the level of intracellular membranes. And the first application that I developed was to modulate the level of phosphatidylinositol 4,5-biphosphates at the level of the plasma membrane. In order to do that, I fused a 5-phosphatase domain to one of the components of the AMAX system, fused to a fluorescent protein to visualize its distribution inside of the cell, and the other component was expressed in the inner leaflet of the plasma membrane. In order to visualize PIP2 inside of the cells, I use a reporter that is based on the pH domain of PLC delta fused to IRFP. When I deliver blue light to the cells, the dimerization between the two Emax components enables to recruit the 5-phosphatase domain at the level of the plasma membrane, where the enzyme finds its substrate, PIP2, and dephosphorylate PIP2 to PI4P. And we can visualize the reaction thanks to the dissociation of the reporter for PIP2 to the plasma membrane. You will see these experiments in the next movie. The next movie will show you the distribution of the PIP2 reporter. This PIP2 reporter is enriched at the level of the plasma membrane in primary neurons before the light irradiation. As soon as the light is turned on, that is highlighted by the white dot in the corner of the movie, you will see the rapid dissociation of this probe from the plasma membrane and its accumulation in the cytosol. When the light is off, you will see the rapid reaccumulation of the probe to the plasma membrane, indicating that PIP2 is resynthesized at the plasma membrane. And this stimulation can be repeated as many times as we want because this system is extremely reversible. One of the most interesting platform of communication inside of the cells is represented by sites of close-up position between the membranes of different organelles. So I was wondering whether I could develop a tool that would enable me to generate or expand this platform of contacts among different organelles. In order to do that, as you can see here in this image, I expressed the two components of my dimerization system on the membranes of different organelles in which I was interested in mediating the dimerization. Upon blue light stimulation, the dimerization between the two photoreceptors would enable me to bring in close-up position the membrane of different organelles, thereby generating and stabilizing these the organelle contact sites. This system is extremely powerful, and as you will see in the next movie, the first one will show you how we can use these tools to induce membrane contact sites between the endoplasmic reticulum in green that express one of these components and lysosomes that express the other component that are highlighted in magenta. As you will see in the movie, when the light is turned on, indicated by the white dot inside of the movie, you will see an increase in the overlap between the two organelles that is indicated by the increase in the white signal among the organelle. When the light is turned off, we will see a rapid dissociation of the two organelles. This is another example where we develop a tool to induce the contact sites between the endoplasmic reticulum here in green and mitochondria here in magenta. Or for instance, like in this case, Chilun Chang at St. Jude Children's Hospital is using this tool to induce contacts between the endoplasmic reticulum and the plasma membrane. As you can see here, where we can see the arrowhead, upon light irradiation, we can see the formation of this enlarged and more stable association between these two organelles, where we can really modulate and study their dynamicity. So at this point, after I proved that we can really use this system for the rapid and reverse modulation and generation of organelle contact sites, I wonder whether I could develop tools in order to interrogate and study the reaction that occur at these sites. So far, we know that the most well-characterized reaction happening at these contacts are the transfer of lipids. And so I tried to generate a tool for the acute manipulation of the intracellular PI4P via reconstitution of an ER trans-Golgi tether that I called OptoVAP. This tether comprises two proteins, OSBP. OSBP is a protein that, thanks to its pH domain, can bind membranes where PI4P is enriched. Thanks to its FFAT motif, it can bind the MSP domain of VAB, a protein expressed on the endoplasmic reticulum, and thanks to its ORD domain, it can transfer PI4P from the Golgi membranes to the endoplasmic reticulum where PI4P is degraded. In order to exert light-dependent control over this tether, I expressed a soluble MSP domain of AB fused to a fluorescent protein for its visualization inside of the cells and to one of the components of the dimerization system, while I expressed the other components of the surface of the ER. The MSP domain of AB maintains its capability to bind endogenous OSBP, but in the absence of activating light, these two components are too far apart, and it is impossible for the OSBP protein to transfer PI4P across the membranes. While when we radiate with blue light the system, the dimerization between the two Emax components regenerate the close proximity of this tethering system between the two membranes of the ER and the Golgi, and you will see how in this case we can actually activate the direct transfer of PI4P from the two membranes. We can visualize the distribution of PI4P thanks to a reporter, IRFP-P4C, and I think that the next movie will show you the power of these experiments and this application. So the next movie is going to show you two images. On the left side of the movie, you will see the signal of the MSP domain of AB. Before light activation, the signal is diffused in the whole cytosol of the cell, and you will see an area of accumulation in correspondence of the Golgi, where there is an enrichment of PI4P because this domain is associated to endogenous OSPP that is enriched at this membrane. On the right side, you will see the signal associated to PI4P, and this, as you will see also, PI4P is already accumulated at the Golgi. When the activating light is turned on and is highlighted by the white dot on the movie, you will see a dramatic association of the MSP domain of a B to the ER in the movie on the right side, and the concomitant reduction of PI4P on the Golgi in the movie on the left. This reduction and accumulation persists as long as the activating light is delivered to the system, but when the blue light is turned off, you will see a very rapid release of the MSP domain of a B from the ER, and a subsequent accumulation of PI4P in the Golgi over a 5 minutes period of time. In order to prove that this PI4P transfer was specifically mediated by our VAP-B OSPP tethering complex, we pre-incubated the cells with itraconazole, that is a drug that specifically occludes the ORD domain of OSPP. And in this case, despite the efficient, as you can see in the graph here, despite the efficient formation of the activation of OptoVAP, we couldn't detect any PI4P transfer. In order to make sure that also we were able to recapitulate VAP-B function inside of the cells, we expressed these tools in HeLa cell lines that were gene edited and didn't express either VAP-A or VAP-B. This cell line is known to have a disrupted Golgi complex with the endosomes enriched with PI4P. We have been able to efficiently express our tool in this cell line, and also in this case, upon blue light activation, we could activate the light-dependent reduction of PI4P from these ER and from these Golgi endosomal elements. And after the pre-incubation with itraconazole, again, this lipid transfer didn't happen anymore. We also confirmed that the PI4P transfer was specifically mediated by the ORD domain OSPP, because when we induced the membrane tethering simply using the pH domain of OSPP without the lipid transfer module, we couldn't detect any PI4P transfer anymore. So with our work, we have engineered a dramatically improved photodimerizer pair that we call enhanced magnets, using a combination of protein design and cellular screening strategies that can be broadly applicable to other reagents optimization. And I hope that I've been able to show you how we have thoroughly validated these EMAC constructs in a range of cellular assay, both in whole cell and local irradiation condition, involving protein recruitment to different membranes, intra-organelle association, and lipid metabolism and trafficking. So I love open science, so all the constructs that I show you today in this presentation and many more are available on Agene, and please reach out to me in the case you would like to use this tool for your biological application. And last but not least, I would like to thank Pietro De Camilli and his lab at Yale, where I perform all this work, and in particularly Aniel and Andres, Jörg Beversdorf and Andrew Barentan, that they helped me in the setup of the microscope system for this type of experiments, and they enable me and they support me with the mathematical model to quantify the blue light-dependent confinement described in the first part of this work. And then I would like to thank Loren Luger at Janillian for his outstanding mentoring in protein engineering, and you all for your attention.